All right, this is going to be a short review on DNA versus RNA. So in our last video, we looked at the structure of DNA, but DNA is not the only nucleic acid. So keep in mind what DNA stands for. So DNA stands for deoxyribose nucleic acid. So nucleic acid is describing the type of molecule that it is. We talked in this class about 4 different types of macromolecules. We've got carbohydrates, lipids, proteins, and nucleic acids. So DNA falls into the category of nucleic acids. So that's where the N and the A part are coming from for the abbreviation DNA. The D part comes from the type of sugar that it has. So it has a deoxyribose sugar. Deoxy means that it's actually lacking an oxygen atom. So what I've drawn here is a deoxyribose sugar so that we can compare it to the sugar that we're going to see in another nucleic acid that I want to look at, which is called RNA. So I've done my best here to note the different sugar here. We've drawn in RNA, and RNA is just another type of nucleic acid. That's the category of macromolecule that it would fall under. You don't need to memorize the structure of the sugar or anything like that. What I would focus on, though, is what gives it its different name, which is the fact that here the DNA deoxy, it's lacking in oxygen that the ribose sugar does have. So this is a ribose sugar. And over here, this is a deoxyribose. And that's one of the major differences between DNA and RNA is the type of sugar they have. Now let's take a look at another difference. All right, what I've done my best to draw here is a couple of other differences between DNA and RNA. Probably the most noticeable here is the fact that DNA is what we call double-stranded. It's got 2 strands here of these strands of nucleotides all stuck together, whereas RNA is what we would consider single-stranded. So this is single-stranded. Another big difference are the options that they have for nitrogenous bases. So in this case, we're used to kind of our T, A, G, C. Those are your options over here. If we look at RNA, it still has an option for adenine, still has an option for guanine, and still has an option for cytosine. However, it doesn't have thymine as a nitrogenous base option. Instead, what you have here is uracil. So that's what makes RNA unique, are the options that it has for nitrogenous bases. And then as we discussed before, the difference is the type of sugar. So all of these sugars are ribose sugars, so I'm going to label them with an R. And all of these are deoxyribose sugars. I'm labeling all of these with a D. You'll notice that they do still have quite a bit in common. They're still made up of nucleotides. So let's remind ourselves what a nucleotide is. A nucleotide is a phosphate, a sugar, and a nitrogenous base. So both of these are still made up of nucleotides as the monomers, those building blocks. And those nucleotides do still have the same components of the phosphate, the sugar, and the nitrogenous base. It's just a matter of what type of sugar and what type of nitrogenous base you have. So there you have it, major differences between DNA and RNA. That's going to be important as we move forward and we look into the different types of RNA, which are mRNA we're going to explore. We're going to look into tRNA, and we're also going to look into rRNA. So those are some different versions of RNA that we're going to explore next.